The cell squeegee, technically called a cell scraper, they look just like mini squeegees that you would use to clean your car window, except here you can use them to scrape cells off of a dish, and it's really helpful when you're trying to like collect cell lysate, so like break open cells and see what's inside, maybe do a Western blot to see what proteins are being expressed, do some RNA sequencing, all sorts of different things that you can do, and this cell scraper is going to help you get your cells off, and so you can basically, you'll actually be able to see as you're looking at it, you'll be able to see when you go down, you can see like the cells will get scraped off into this bottom portion, and then you can just, if you have a pipette, you can just collect your cells like this. Your cells will probably be dead, even if you don't use a lysis buffer. But often you are using the lysis buffer at this step. But basically, this is a harsh method, and so most of the time, there are some exceptions, but most of the time you're going to be using this if you don't care that the cells survive. If you're caring that the cells survive, say you're trying to passage the cells, so maybe they're starting to overgrow on one plate, and so you need to dilute them and plate them on more plates. Then you want to use something gentle. You typically use some sort of like enzymatic method, so that uses enzymes such as trypsin to cut apart the protein-protein connections that are keeping the cells the cells like glued to one another and to the dish, the specially coated dish. So there's these proteins called like cadherins as well as other proteins. The cadherins are dependent on calcium, and so EDTA is a chelator. It's a metal binder. It'll bind to that calcium and steal it, so the proteins can't make those connections as strongly, and then the trypsin can go in and as a protein chewer, it can sever those connections, and it can sever the connections between cells and between cells in the plate without actually killing the cells. Hopefully, that's why one of the reasons why you add the EDTA is so that you don't have to add too much trypsin, because if you add too much trypsin, it'll start chewing up other stuff, or chewing up excessively, and we don't want that, so we limit it. But basically, with trypsin, you're able to then like reuse the cells, and you can use trypsin and detach cells just like you would normally, like we talked about yesterday, if you want to then lyse the cells later. But often what you're doing is you can actually lyse the cells in the dish, and whether or not you lyse them in the dish, you can use a cell scraper because you don't care if the cells survive, if all you care about is what's inside the cells and you're about to break them open anyway. But you do want to make sure that you're breaking them open into the solution that you want. So this first step typically is to remove whatever media it was growing in. Just like we talked about yesterday with the trypsin, you're going to remove the media and then typically do a wash with PBS. But in this case, you can use like normal PBS. It's okay that it has those salts in it because we don't, like the metals, the calcium and the magnesium. It's okay that they're in here now because we don't care about the EDTA or the cadherins doing their thing, because we're basically just going to mechanically disrupt the connections. And so you do a PBS wash. One of the things is when you're doing these washes, you want to make sure that you're not, you don't want to just like squirt directly on the cells, because that's going to like detach the cells, and you don't want to detach them yet because you want to wash them off first. You want to wash off all that media stuff. So what you can do is just pipette like down on the edge, and when you pipette down on the edge slowly, then you can kind of just rock the liquid over the surface of the cells. It's not doing it well now because there's not actually any cells on here, but when there are cells in there, it'll like kind of glide over the cells. You want to wash it all completely, then tilt the plate and collect the liquid. If you're using like a larger volume and you're using one of those serological pipettes, it's still good to, those often leave a little bit, and so then you want to like keep your plate tilted, let everything go to the bottom, and then section it off, and then so that's just your wash, and then you can do your lysis buffer. Now, because you are going to collect the cells here, it's okay if you disrupt them. So it's not like you can squirt directly on the cells now because you're trying to kill them and get them off the plate into the liquid. But if you detach the cells before, then they're going to come out into the liquid that you're not trying to get them out into. So when you do the lysis buffer, you want to make sure that it gets spread evenly, and so typically I just like drop-wise add it, or that's not very drop-wise, it's hard to do this on camera, but kind of just like drop-wise add it over the plate, and then tilt and let it all go. And then some of the protocols you might let it sit for various times. Some things you might do it right away. But you want to make sure that when you're scraping the cells, you're getting them all. So you can look at look in a light, and you should be able to see the cells come off. And so make sure, especially the edges, you want to get it, and try to don't go up, down, up, down, up, down, because then you're just going to be spreading it to the parts where you're not going. Try to keep going down towards the liquid that's pooling and collecting. Once you collect all this liquid, if there's still some cells stuck around, you can often take a little bit more of your lysis buffer and kind of wash them down, or use some of the cell solution, but then you can have more cells get stuck in the process of doing that. And yeah, so then you can do whatever you wanted to do with it. And so you can do something similar with just a PBS or something, some sort of buffer that doesn't have the lysis stuff. So with the lysis buffer, those typically have a detergent. So these artificial soaps, that's going to help break things open. The different protocols are going to have different intensenesses of the detergent, their concentration wise, or how harsh those detergents are. Depending on how harsh the detergents are, they're better or worse at totally breaking things open, totally lysing things. If you're using milder conditions, sometimes you want to do this if you want to keep protein connections, see what's binding what, and that sort of thing. You'll use a milder condition, but then you're probably going to have to use some sort of mechanical disruption to actually finish the lysis. So often this is done with triturating, so pulling the cells up and down through a needle. Homogenizing, there's different tools that you can use to grind things and all sorts of stuff to break things open. If you're using really harsh detergent conditions, then the cells should just be lysed already, and you don't have to go through those other steps. But yeah, so that's the basics. So we can call this in-dish lysis if you just do it on here, or you can scrape off the cells, get them in a tube, and then do the lysis in there. Once the cells are broken open, then you're typically going to spin them down to get rid of all the gunk, the membraney stuff that's not soluble, and then you can do whatever you wanted with that lysate. So yeah, so that is the basics. And cellular scrapers, come on, let's just call them cell squeegees. They're cell squeegees. Awesome.